Good afternoon. Welcome you all to the today's presentation topic Insect Body Organization. Insect body organization refers to the structural arrangement of an insect body parts. Structural arrangement of an insect body parts mean how the insect body is divided into three main regions or part, that is head part, thorax part and abdomen part, and what are the appendages are present in each part, such as antenna, legs, wings, etc., and how these parts and appendages work together to help the insect live, move, feed and reproduce is the structural arrangement of an insect body parts. So in this session we are discussing in detail body segmentation in insects, structure of head, Sclerites and sutures of head, types of insect heads, insect neck or cervix, thorax and its appendages, abdomen and its appendages. Body segmentation in insects. The insect's body generally consisting of totally 20 segments, and these segments are grouped into three distinct regions, that is one head region, thorax region and abdominal region. Head region consisting of 6 segments, thorax region consisting of 3 segments and abdominal region consisting of 11 segments. So totally the insect body generally contains 20 segments. So head bears appendages such as eyes, antenna and mouth parts. Thorax, it is divided into prothorax, mesothorax and metathorax, and bears appendages such as 3 pairs of legs and 2 pairs of wings. Abdomen. Abdomen contains genital appendages and these are typically located on the ventral side of 8th and 9th abdominal segments. Then what do you mean by tagmata and tagmosis? Tagmata is equal to a region. Insect body is divided into 3 regions, that is head region, thorax region and abdominal region. Region is equal to tagmata, therefore the insect body is divided into 3 tagmata, that is head tagmata, thoracic tagmata and abdominal tagmata. The insect body is differentiated into 3 distinct regions is known as tagmata, that is head tagmata, thorax tagmata and abdomen tagmata. Each tagmata is composed of a specific group of body segments, that is head tagmata contains 6 segments, thorax tagmata contains 3 segments, abdominal tagmata contains typically 9 to 11 segments. So here 6 segments in the head. These 6 segments are grouped into a region. In thorax, these 3 segments are grouped into a region. In the abdomen, 9 to 11 segments are grouped into a regions. That is, the grouping of body segment into a functional region is known as tagmosis. The grouping of body segment into a functional regions is known as tagmosis. Then segmentation type, that is primary segmentation and secondary segmentation. The arrangement of body segment during the embryonic stage is known as primary segmentation. The modified arrangement of segments in the adult stage. The modified arrangement of the segment in the adult stage is referred as secondary segmentation. So structure of insect head. Insect head is the anterior-most tagmata of the insect body. It is a highly specialized and sclerotized structure. Sclerotized structure means it is made up of or made with hard cuticular plates, and head play a very important role in sensory perception and feeding. See here head composition. Head capsule is a sclerotized structure, meaning it is covered with hard cuticular plates called sclerites. This head is articulated with the several appendages such as antenna, various mouthparts and eyes, which are crucial for sensory and feeding function. When the head capsule is considered without appendages and contain only sclerite, that referred as cranium. Head capsule without appendages is known as cranium. The insect head is derived from six embryonic segments. See the region of heads here: procephalon and gnathocephalon. Anterior part of the head is known as procephalon. It contains eyes and antenna, and it is responsible for sensory functions. Gnathocephalon. This is the gnathocephalon region. So this is the posterior part of the head that bear mouth parts, therefore it is responsible for feeding functions. Next, see here head appendages. Any attached structures to the head are known as head appendages. The head consisting of following appendages, that is eyes, antenna and mouth parts. Eyes include compound eyes. These are useful for image formation. Simple eyes in between the compound eyes. These are useful for light intensity detection. Next appendage is antenna. These are the paired segmented structure used for olfaction, touch and hearing sensation. And other appendages of head are mouth parts, that is labrum, upper lip, mandible, jaw-like structure for cutting and chewing the food particles. Next is the maxillae, paired structure with sensory and food handling roles. Labrum, this is the lower lip. Epipharynx, this is associated with the labrum and sensory in function. Hypopharynx, this is the tongue-like structures, help in food manipulation and salivary duct passage. Functions of the head. Head help in feeding due to presence of specialized mouthpart. Sensory perception with help of eyes, antenna and palps, and coordination of the responses due to the presence of brain in the head. Next, sclerites and suture of insect head. Head capsule is formed by the union of several cuticular plates. See, this is yellow colored cuticular plates, light yellow colored cuticular plates, then blue color plates, red color, these red color mandibles are there. So such several plates are there, such several cuticular plates are there. The head capsule is formed by the union of several cuticular plates are known as sclerites, and these sclerites are get jointed together. The sclerites are joined together by cuticular line or ridges is known as sutures. A suture is a linear invagination of exoskeleton between any two sclerites. So major sclerites of insect head, that is the labrum, next is the clypeus, Next is the frons. This is the largest sclerite of insect head, form the facial part of the insects, then vertex, the top portion of the head, located above the frons, and the area between the two compound eye is known as vertex or epicranium. Next is the zena sclerite, the region below the compound eyes, extended just above the mandibles, are known as zena. Next, ocular sclerite, the ring-like cuticular structures. The ring-like cuticular structure present around each compound eyes are the ocular sclerites. Next, antennal sclerites. This provide base for antennae, surround the scape part of the antenna, and it is well developed in Plecoptera, that is stone insects. Next is the occiput. This is present in the backside, an inverted U-shaped structure situated between the epicranium and post-occiput is the occiput. Then last sclerite is the post-occiput. It is their backside of the cranium, the extreme posterior part of the head, located just before the neck region, is the post-occiput. All the above sclerites are interconnected by cuticular ridges or sutures, and these sutures serve as site for muscle attachment within the head. Next, we are discussing about sutures of the insect head. The cuticular plates of the insect head are joined together by cuticular lines or ridges known as sutures. So see here important suture of insect head, that is epicranial suture, ocular suture, genal suture, antennal suture, epistomal suture, clypeolabral suture, occipital suture, and post-occipital suture. So epicranial suture, it is an inverted Y-shaped suture on the top of the head. It consisting of two lateral arms known as frontal sutures and a central vertical stem is known as coronal suture. This epicranial suture, also known as ecdysial suture or line of weakness, because the exuvia split along this suture during the process of molting. Next, ocular suture, a circular suture that encircle each compound eyes is known as ocular suture. Next is a genal suture located on the lateral side of the head, making the boundary of the gena is the genal suture. Next, antennal suture, a marginal depressed ring that surround the antennal socket is the antennal suture. Next, epistomal suture, a transverse suture present between the clypeus and the frons is the epistomal suture. Next is the clypeolabral suture found between the clypeus and labrum, located on the lower margin of the clypeus from which the labrum hangs downwards. Next is the occipital suture, the U-shaped or horseshoe-shaped suture located between the epicranium and the occiput. Epicranium and occiput is the occipital suture. Post-occipital suture. So it is there here, post-occipital suture. This is the only true suture in the insect head, located at the posterior end of the head, and it's demarcate the head from the neck region. It separates the head from neck region. That's why we are calling post-occipital suture as the real suture of the insect head. Next, types of insect heads. Based on the inclination of long axis of the head, 1 point, and the orientation of the mouth parts. 1 point is the inclination of the long axis of the head, and the another point is the orientation of the mouth parts. The insect heads are classified into three types. That is one hypognathous head. Hypo means below, gnatha means jaw. So here the long axis of the head is vertical and it is at right angle to the long axis of the body. Here long axis of the body of insect and here long axis of the head is vertical. So here long axis of the head is vertical and it is at right angle to the long axis of the insect body, and the mouth parts are ventrally placed and projected downward. See here. So this type of mouth part is known as hypognathous. This type of head is known as hypognathous head. This is also known as orthopteroid type head. Why? Because these are present in mostly present in Arthroptera order insects such as in grasshopper, locust, cricket, and mole cricket. Next is the prognathous head. Pro means in front, gnathous means jaws. The jaws are directing in forward direction. Here the long axis of the head is horizontal and it lies in the line with the long axis of the body, and the mouth parts are directed forwards, and this type of mouth parts are found in the beetles. That's why we are calling this mouth parts are directed forwards, and this type of head is found in the beetles, and this kind of head is also known as coleopterite type of head. Next, opistognathous head. Ophisto means behind, gnathous means jaw. See here, the head is deflexed, mean that bent or curved backwards, and mouth parts are directed backwards and held between the forelegs. This type of head is there in stink bug and red cotton bugs. That's why we are calling this type of head as the hemipterite type of head or ophisthodyngus type of head. Next part is the neck or cervix. The head is connected to the thorax by means of a membranous region is known as neck or cervix. The cervical membrane is flexible, allowing the free movement of the head. Next region is the thoracic region or thoracic tagmata. This is the second or middle tagmata of the insect body. It is highly musculated region and responsible for locomotion. It is divided into three segments, that is prothorax, mesothorax, and metathorax. Sclerite of each thoracic segment, see here. Dorsal sclerite are known as notum or tergum. Lateral sclerite are known as pleura, and ventral sclerite are known as sternum. See here appendages of thorax. Thorax consisting of two pairs of wings, that is present one pair on mesothorax, that is forewings, and another pair on metathorax, that is the hind wings. Due to the presence of wings on meso and metathorax, this meso and metathoracic segments are known as pterothorax. These two segments together are known as pterothorax. Three pairs of legs. So one pair on each thoracic segment, that is foreleg, mid leg, and hind leg. Two pairs of spiracles also there, located on the pleura of mesothorax and metathorax, and thorax is mainly concerned or primarily responsible for locomotion due to the presence of wings and legs in them. Next tagmata or region is the abdomen. The third and posterior tagma of the insect body is the abdomen. It is composed of 9 to 11 segments called as uromeres. The abdomen is highly flexible and which is essential for copulation and oviposition in insect. Each abdominal segment consisting of a dorsal plate is known as tergum or notum and ventral plate known as sternum. There is no pleural plate in the abdomen. Therefore, the spiracles are located on the dorsolateral extension of the tergum. In grasshopper, there are 8 pairs of abdominal spiracles located on the first 8 abdominal segment, along with the pair of tympanum on the first abdominal segment. So, first abdominal segment consisting of tympanum also. This is the hearing organ. At the same time, a pair of spiracle also there in first abdominal segment. Abdominal appendages in adult insects. Abdominal appendages include genital organs and cerci. Here, cerci is there at the last abdominal segment. It serves as a sensory function. The eighth and ninth abdominal segment bear the female genital structure, while the ninth segment bears the male genital structure. Function: the abdomen is mainly concerned with the reproduction and metabolism. See here abdominal spiracle in grasshopper. These are the respiratory openings present on the ventral side of insect body. In mesothorax, one pair; in metathorax, another pair; and in abdominal region also there up to 8 pair in grasshopper, and it's differ from insect to insects. See here clearly about female genital structure. The ovipositor typically consisting of 2 valvi for 1, 2, and which gives rise to 3 pairs of valvulae. See here, from the base of the eighth abdominal segment, valvifer 1 will arise, and this valvifer gives 1 pair of valvulae. From the base of ninth abdominal segment, valvifer 2 will be arise, and it gives rise to 1 pair of valvulae 2 and 1 pair of valvulae 3. Totally 3 pair of valvulae are there, which are originated from 2 pairs of valvifers. Male genital structure. The entire copulatory organ of higher insect is generally called as adagus or edagus. In addition to its role in insect insemination, it may also assist in clasping, that is, catching and providing sensory stimulation to the female. Here, clasping means it's handling the female and provide sensory stimulation to the female. Typical components include a median tubular penis is there, often referred as adagus, and inner tube of this penis is known as endophallus, and which is averted during the process of insemination. And the ejaculatory duct open at the gonophore, which is located either at the tip of the penis or at the end of the endophallus. So additional structures such as on either side of penis, a pair of lobes are known as parameres also there. They mainly serve as clasping or sensory in function. Thank you. That's it in this session. In the coming session, we are discussing about abdominal appendages in wingless insects, immature winged insects, and in adult insects. Thank you. Cuticular appendages. Cuticular appendages are the structures that extend from the insect exoskeleton or the insect body wall. So these are of two types based on their structures and association with the epidermis. C, that is one non-cellular appendages, and second one is the cellular appendages. Non-cellular appendages mean these are not produced by the epidermal layer, but cellular appendages, these are produced by the epidermal cells. That is the difference. So, non-cellular appendages, these appendages do not have any direct association with the epidermis. They are rigidly attached with the cuticle but are not formed from the living cell. That's why they are non-cellular. So example for non-cellular appendages is the minute hairs and thorns. So these hairs are sensory in function. So they detect this hair-like structure detect air movement. So they are sensory in function, and the thorns are defensive in function for protection. Therefore, these are the non-cellular appendages. Next, we are discussing about cellular appendages. These appendages are directly associated with the epidermal layer, meaning they are formed by the epidermal cells. These are of two types, unicellular and multicellular cellular appendages. Unicellular appendages formed from the single epidermal cell only. When two or more epidermal cells combinedly gives structures, that is the multicellular structure. So see here the example, CT and gland are the example for the cellular appendages. These appendages are functional and may serve in sensation, defense, communication and production of secretions. In the coming slide you will see examples. So because they are cellular, they have living component and can grow, regenerate and sometimes secrete substances. Why? Because they are cellular and living. That's why they can regenerate, they can produce any substances also. See here cellular appendages, unicellular appendages and multicellular appendages. Unicellular appendages are clothing hairs and plumose hairs in honeybee. Bristles are there in flies. Scales are there in butterflies and moths. Glandular seta are present in caterpillars. Sensory seta are present in many insects. Seta are present in many insects. Then after multicellular cellular appendages, that is movable spurs and immovable spines. See here images. Clothing hairs or plumose hairs are there in honeybees and bristles are there in flies. Then after scales are there in moths and butterflies. Then after glandular seta are, see here, these are the glandular seta present in the caterpillar. Sensory seta are present in many insects. Seta are hair-like outgrowth present in many insects. Seta, this is a hair-like outgrowth. Epidermal cell which produce seta are known as trichogen cell. Trichogen, it generated trichome. That's why the name trichogen. The socket forming cell around the trichogen is known as termogen. Socket forming housing for trichogen is the termogen. The study of arrangement of the seta is known as cuticular. Study of arrangement of the seta is known as cuticular. In some dense arrangement is there, in some space arrangement is there. The study of that arrangement is the cuticular. And multicellular spurs, these are the spurs movable structures, and spines, these are the immobile structures. Then after wax gland, this is also multicellular structures present in honeybees and mealybugs. Next, lag gland, this is present in lag insect multicellular structures. Molting gland is present in many insect, they secrete molting fluids. And androconia or scent cell present in the moths. And then after poison gland is present in the slug caterpillar. This is also epidermal appendage. This is also cuticular appendage, that too multicellular. So that's it from my side. Thank you. Welcome you all. In this session, we are discussing abdominal appendages in wingless insect, immature winged insects, and adult insects. Before going into the discussion, what is the appendage? Appendages are the parts that are attached to the main body part. Any parts which attached with the main body parts are the appendages. For example, an insect body is divided into three regions: head region, thorax region, and abdomen region. Head region also consisting of attached part, that is appendages such as antenna, eyes, and mouth parts. In thorax region also appendages are there, that are the wings and legs. Like that, in abdomen also appendages are there. We are discussing in the session that abdominal appendages only. So abdominal appendages in wingless insects. What are the appendages are there in wingless insects? They are styli, colophore, retinaculum, and furcula. These are the abdominal appendages are there in wingless insect. I think you may know that what are the wingless insects order, that is Thysanura, Procura, Diplura, and Columbola. These are the wingless insect order. So this order insects, they contain the following abdominal appendages such as styli, colophore, retinaculum, and furcula. We will see one by one in the coming slide with the images. So abdominal appendages in wingless insects. In wingless insects, abdominal appendages are generally pregenital, mean they are present before the genital segment, and they perform various functions. Let's see what are their function one by one. That is, first abdominal appendage in wingless insect is the styli. See here, this styli will found in the silverfish. This is the image for silverfish. And the styli are there on the ventral side of the abdomen. These are the styli. Styli are paired, tube-like or peg-like outgrowth. See, these are the tube-like or peg-like outgrowth present on the ventral surface of the body. They are believed to be reduced abdominal legs. Then, what is their role? So functionally, they assist in locomotion and stability during the movement. That is the function of the styli. So styli are there in silverfish. Silverfish are belong to the order Thysanura. So next abdominal appendage is the colophore, retinaculum, and furcula. All these abdominal appendages are there in the columbola, that is the springtail. See, what is the colophore? Colophore, also known as ventral tube or glue peg. See here, light brown or red color structure is there now, that is the colophore. It is there in columbola, that is springtail insects. It is located on the ventral side of the first abdominal segment. This is the head, this is the thorax, and this is the abdomen. On the ventral side of the first abdominal segment, we can see colophore. So it is a cylindrical structure and it can be produced outward by the hydrostatic pressure of the hemolymph, that is the blood of the insects. It is so has an organ of adhesion, mean allow the insect to stick to the surface of the substratum. It also helps in water absorption from the substrate and may also play a role in respiration also. These are the function of this colophore or glue peg, help in adhesion, water absorption, and respiration. And next structure or next appendage is the retinaculum or tinaculum or catch. See here, this green color structure is there now. That is the retinaculum or tinaculum or catch. It is also there in Columbola springtail insect only. It is located on the ventral side of the third abdominal segment. This is the first abdominal segment and this is the third abdominal segment. On the ventral side of the third abdominal segment, we can see retinaculum in case of Columbola. So it acts as a hook-like structure and hold the furcula. This is the furcula and it's held by the hook-like structure, that is the retinaculum or tinaculum. It holds the furcula in a locked position when this is in not use or when this is in not function. Prevent unintentional release of the jumping mechanism. It prevents the unintentional movement of this spring tail. The structure is a retinaculum or tenaculum. It will prevent the unintentional movement of this spring tail. Next structure is the furcula or furca. So this is the furcula or furca. It is also there in columbola or spring tail. It is located on the ventral side of the fourth abdominal segment. So this is a Y-shaped forked structure. It is kept in tensed position, mean held tightly under pressure under the body by the retinaculum. It is held by means of the retinaculum. This is the previous structure we already discussed. When released, it strike against the substratum and propel the insect into the air. So it is act as jumping organ. So allowing it to jump, and it is a unique locomotory adaptation in the spring tails. So this is the organ which help this insect to move into the air. That's why the name springtail. It possesses spring-like structure. That is the furcula or furca. That's why the name springtail. So these are the only abdominal appendages in wingless insect. Next, we are going to discuss about abdominal appendages in immature stages of winged insects. In winged insects, immature stages, we can see anal papillae, tracheal gills, dolichasters, and then prolegs. So abdominal appendages in immature stages of winged insects. In the immature stages mean that is the larvae or nymph. The larvae or nymph stage of winged insect possess several abdominal appendages that help in respiration, locomotion, or physiological regulation. We will see in detail about that appendages along with their function in the coming slide. So these structures are not found in the adult stage. That's why the heading here is the abdominal appendages in immature stages of winged insects, but are very important for the larval and nymphal survival and development. They are very important for their survival and growth. So first one is the anal papillae. So this found in the larvae of the mosquito also known as wriggler. They found around the anal region. See their structure and function. Typically, there are four finger-like projections are found surrounding to the anal region or the anal papillae. They are involved in osmoregulation. That is, they regulate salt and ion concentration in aquatic environment. Next is the tracheal gill. Tracheal gills are there in the needs of Odonata and Ephemeroptera. So Odonata example is the dragonflies and damselfly. Ephemeroptera and other insects are the mayflies. So tracheal gills we can see in the needs of Odonata and Ephemeroptera. Function of the tracheal gill is the tracheal gills are the lateral or terminal extension of the body. See here, these are the terminal extension or lateral extension of the body that are richly supplied with the trachea. That's why the name tracheal gills. They help in extracting dissolved oxygen from the water. They help in absorption of dissolved oxygen from the water. Types of tracheal gills. So lateral gills are there in mayfly. Caudal gills are there in damselfly nits, and rectal gills are there in dragonfly nits. So lateral gills found has 7 pair on the lateral side of the abdomen. See 1 pair, 2 pair, 3 pair, 4 pair, and 7 pairs are there in the mayfly nits. Caudal gills are there in damselfly nits. 2 or 3 leaf-like structures. See 1, 2, 3 leaf-like structure at the end of caudal end. They are also help in swimming apart from the respiration. Help in respiration as well as in swimming also. A rectal gill. They are present in the dragonfly nits. That is the immature stage of dragonfly. Gills are located internally in the rectal chamber. That's why we are not seeing here. They are present internally. Water is pumped in and out of the rectum, facilitating gas exchange. They help in respiration only. So next structure is the dolichasters. Dolichasters are there in grubs of antlion. So these are the dolichasters. Here see, small minute structures. Dolichasters are the segmental protuberance. This segment consisting the protuberance, this segment contain protuberance. These are the segmental protuberance found on the abdomen. Each is fringed with the setae. Each protuberance is fringed with the hair-like structures. They may assist in sensing environment. So they can sense the environment by using this dolicaster and aid in movement through the soil or substrate. They also help in a movement of this insect through the soil or substrate. And next, abdominal appendage in immature stage of winged insect is the prolegs. See, these are the true legs and these are the prolegs. So these prolegs are there in caterpillars, loopers, semi-loopers, and in softfly larvae also. So see here structure and function. These are the unsegmented, fleshy and thick appendages. There is no segments. They are feel fleshy and thick appendages, normally present 2 to 5 pair on the abdominal segment here. 1, 2, 3, 4, 5 pairs are there. The tip of each proleg is a plate-like structure. Tip of each proleg is plate-like structure. We call that plate-like structure as the planta. And this planta bears crotchets. That is, the tiny hooks are there. So that plate-like structure bears small hook-like structures, that is the crotchets. These crotchets help the larvae in crawling and gripping the surface effectively. That is the importance of crotchets in case of prolegs.
Abdominal appendages in winged adult insects. These appendages play diverse roles such as protection, respiration, reproduction, sensory perception, and defense. So, first appendage in winged adult insect is cornicle. These cornicles are there in aphids. These are also known as siphonculi. These are the pair of tubular projections. See, this is the pair of tubular projection located on the fifth or sixth abdominal segment. So, what is the role of cornicles? Cornicles secrete a waxy defensive fluid that deters the predators. Next is the caudal breathing tube. Caudal breathing tube is there in water scorpion. It consisting of two grooved filaments that come together to form a tube-like structure is known as caudal breathing tube, and it is there at the end of the abdomen. This caudal breathing tube allows the insect to breathe while they are submerged, and it is act as a snorkel. It functioning like a snorkel. Cerci located on the 11th abdominal segment. These are mainly sensory organs, but they also play other roles such as in defense, mating, or grooming, depending on the species to species. See here, sclerotized or forceps-like cerci is there in airwig. Long and many segmented cerci is there in mayfly. Long and unsegmented cerci are there in cricket. Short and many segmented cerci are there in cockroach. Short and unsegmented cerci are there in grasshopper. Asymmetrical cerci are there in male embid, that is Embryopteran web spinner insects. Next is the median caudal filament. This median caudal filament found in the mayfly and silverfish. It is a elongated epiproct forming a central filament between the cerci. So this is also a sensory structure. Next is the pygostyle is there in squalid wasp, a pair of unsegmented cerci-like structure. At the end of the abdomen in squalid wasp is the pygostyles. They involved in mating or oviposition support. Next appendage is the anal styli. They are there in male cockroach only. They found at the end of the abdomen. These are the short, unsegmented, paired structures. Help in holding the female during copulation. Ovipositor, an egg-laying organ found in the female insect is the ovipositor. These are the highly modified based on the insect ecology. See here, long and sword-like ovipositor is there in catidid, a long-hand grasshopper. Needle-like ovipositor is there in cricket. Sting-like ovipositor is there in worker honeybees. Pseudo-ovipositors. Here there is no particular appendage. In this case, the body parts are developed into the ovipositor, so that's why the name pseudo-ovipositor. This kind of ovipositors are there in fruit fly and housefly. See, in fruit flies, the sharp pointed terminal abdomen developed as the pseudo-ovipositors that pierces the fruit skins and lays eggs there. In houseflies, telescopic abdominal segments help in oviposition. They are also known as ovitubes or oviscapt. Male genitalia, external reproductive organs located on the ninth abdominal segment in male insect is the male genitalia. In most insects, these are used for mating and sperm transfer purpose. See here, unique case in dragonfly and damselfly, the functional copulatory male organ is there on the ventral side of the second abdominal segment. See here certain modification in abdominal segments. In springtail, that is columbola, only 6 abdominal segments are visible. In housefly, that is in Musca domestica, only 2 to 5 segments are visible and 6 to 9 segments are telescoped internally. In ants, bees, wasps, first abdominal segment which is fused with the metathorax, prothorax, mesothorax and metathorax. The first abdominal segment which is fused with the metathorax is known as propodium. Second segment, a narrow waist-like structure is known as petiole, and remaining all other segment is commonly called as the gaster. Thank you.
So, welcome, my dear students. In this session, we are discussing about insect body wall. Insect body wall is also known as integument or exoskeleton. So, what is the other name of insect body wall? Very good. See here, insect body wall. This is also known as integument or exoskeleton. You already know. And this is the external covering of the insect body. And it is derived from the ectoderm. of the embryonic stage. See here, structure of the body wall. The insect body wall consisting of three layers. That is, first layer is the cuticle. Second, this green layer is there now. This is the epidermis, and last layer is the basement membrane. See here also, the first layer is the cuticle, and this second layer is the cellular epidermis, and last layer is the basement membrane. So insect body wall totally consisting of three layers. What are those three layers? Outer non-cellular cuticle, middle cellular epidermis. It is also known as hypodermis. Why the name hypodermis? Because it is present below the cuticle. Hypo means below. So that's why the name hypodermis. And the last layer, that is inner non-cellular layer, is the basement membrane. So we are discussing in detail about outer non-cellular layer, that is the cuticle. From here to here, this is the cuticle. This cuticle is actually secreted by the epidermal layer. This epidermis secrete cuticle, and it is again divided into epicuticle, exocuticle, and endocuticle. Cuticle is divided into three layers, that is epicuticle, exocuticle, and endocuticle. Then what is epicuticle? Epicuticle, this is the outermost non-chitinous layer. Here there is no chitin in this layer, and this is the thin layer. So pore canal present in the exocuticle. In exocuticle, pore canals are there that will secrete this epicuticle. Actually, entire cuticular layer is secreted by the epidermal layer. So this epidermal cells gives pore canals. That pore canals secrete one of the layer, that is the epicuticle. This epicuticle is there now. It is again divided into 5 layers. Cuticle consisting of 3 layers, that is one epicuticle, exocuticle, and endocuticle. Epicuticle again consisting of 5 layers. See that 5 layer here. The first layer is the cement layer, second layer is the wax layer, third layer is the cuticulin layer, fourth layer is the outer epicuticle, and fifth layer is the inner epicuticle. So here this epicuticle consisting of totally 5 layers. Cement layer, it is made up of lipids and tanned proteins. Lipids and tanned proteins. What is the role of cement layer then? It is the outermost layer. It protects the wax layer from physical abrasion and environmental factors like rain and heat. So this is the role of cement layer. Then what is the role of wax layer? This is packed with the wax molecule. See, this is the wax layer. So this form a waterproof barrier and prevent the insect from drying out. That is, it prevent the insect from desiccation. That is the role of wax layer. Below the wax layer, cuticulin layer is there. So this is the cuticulin layer. It is the polymerized lipoprotein. It is made up of with polymerized lipoproteins. And what is the role of this cuticulin layer? It is act as a foundation for wax and cement layer. It forms the base for about 2 layers, that is cement and wax layer, and it's block water and chemical from entering inside the insect body. Sometimes we are spraying insecticide, so these are not directly enter into the insect body due to the presence of this cuticulin layer. Yes. Next, outer epicuticle. This is very thin membrane like a layer. It binds cuticulin to the upper layers. The role of outer epicuticle is the binding. It keep all the layer in bind. Next is the inner epicuticle. It contains wax filament and guide and hold wax molecules. So wax layer will be formed in later. So before that, wax molecules are holded in this layer only, that is in inner epicuticle. So it receives wax from the pore canals and ensure even deposition onto the wax layer. later wax layer will be formed. Now, even distribution will be done by this epicuticle only. So this is the information about epicuticle. Epicuticle consisting of totally 5 layers and their roles we already discussed. That is cement layer, wax layer, cuticulin layer, outer epicuticle and inner epicuticle. So after epicuticle, next layer is there, and that is exocuticle. I already told you that in cuticle 3 layers are there, that is epicuticle, exocuticle and endocuticle. About epicuticle we already discussed. Now we are going to discuss about exocuticle. Exo. So exocuticles lies between the epicuticle and endocuticle. So this is the layer that is present in between the epicuticle and endocuticle. So it is made up of chitin and sclerotin. These are kind of proteins. This form the exocuticle layer. So this is a dark color, rigid and hardened layer of the cuticle. Next, last layer of the cuticle is the endocuticle. This is the innermost and thickest layer, and it is made up of chitin and arthropodin. What is chitin and what is arthropodin, we will discuss in the coming slides. And this is the colorless, soft and flexible layer. So we are calling exo and endocuticle combinedly has the procuticle. Procuticle equal to exocuticle plus endocuticle. Why we are calling it procuticle? What is the meaning of procuticle? Procuticle mean this is the main or principal part of the cuticle, and it forms the bulk of the insect's exoskeleton. That's why its name procuticle. So in cuticle 3 layer we discussed epicuticle, exocuticle and endocuticle. Next layer of body wall is the epidermis. So before going into the discussion about epidermis, we will discuss composition of the cuticle. That is, what is chitin, what is arthropodin, what is sclerotin, and what is resin. So chitin, it is a tough nitrogenous polysaccharide made up of a repeating unit of N-acetyl glucosamine. N-acetyl glucosamine, N-acetyl glucosamine, several N-acetyl glucosamine combinedly form chitin. So what is the function of this chitin? Main component of the insect exoskeleton and provides structural support, rigidity, flexibility and protection to the insects. Without chitin, there is no rigidity in their body wall and there is no protection for the insects. So next, arthropodin. See here properties of chitin. This is water insoluble but soluble in dilute acids, alkaline and organic solvents. Next, come to the arthropodin. It is a protein, untanned protein which is water soluble. So what is the function of arthropodin in the insect body wall? It is essential for cross-linking of the chitin molecule. That chitin molecules are cross-linked by this arthropodin only. It forming a strong matrix for mechanical support and protection. So contribute to the toughness and flexibility of the insect cuticle. Arthropodin contribute to the toughness and flexibility of the insect cuticle. Next is the sclerotin. A water insoluble tanned protein is the sclerotin. It play a key role in hardening of the cuticle, making rigid and protective. So contribute to the stiffness and resilience of the insect body. This is because of sclerotin only. Next is the last protein, that is resin. It is an elastic protein found in areas requiring flexibility such as joints. Here resin may be there at joints, wing hinges and some part of the other exoskeleton. The function of resin is it provides elasticity and flexibility, allowing spring-like movement in the insect body. Responsible for resilience, enabling insect to jump, fly and bend their exoskeleton without breaking. This is possible because of this resilient protein. So these are the composition of the cuticle. Cuticle contain proteins such as chitin, arthropodin, sclerotin and resilience. And their roles we already discussed. So we already discussed cuticle. Cuticle consisting of three layers: epicuticle, exocuticle, endocuticle. Now we are moving towards the next layer, that is the middle layer. This is the cellular layer. It's known as epidermis or hypodermis. Single layer of cells are there between the cuticle and basement membrane. The cells of this epidermis are held together by means of desmosomes. These are the cytoplasmic processes. All the epidermal cells are glandular and secrete cuticle and enzymes. I already told you that cuticle is secreted by the epidermal cells only. So here see types of epidermal cells based on their function. For example, you consider this cell has the dermal gland, this cell has the trichogen cells, this cell has the molting gland, this cell has the peristigmatic gland. All the cells of the epidermis are glandular, you know. So what is the role of dermal gland? Dermal gland produce cement layer. Dermal gland produce cement layer. Next is the trichogen cell. Produce hair-like structure on the body wall. Next is the molting gland. This secrete molting fluid which is very important during the process of molting in insect. We will discuss about molting in the next session. Next, peristigmatic gland. These glands found around the spiracles. Spiracles are the respiratory opening in insects. So this peristigmatic gland found around the spiracle and dipteran larvae. These are hydrophobic in nature. So these do not allows water entry through the spiracle inside the insect body. So then what is the function of the epidermis? Epidermis secrete cuticle, produce the enzyme to digest old cuticle. It is essential during the process of molting. Next, it repair wound and gives external appearance to the insect. So these are the functions of the epidermis. So we already discussed about cuticle, epidermis, and next and last layer is the inner non-cellular layer, that is the basement membrane. See here, this white line is the basement membrane. This lies just below the epidermis and it is amorphous. There is no a particular form for this one, and it is granular and composed of neutral mucopolysaccharides, and its thickness is only about 0.5 micrometers. It forms a continuous sheet below the epidermis just like neural lamella. So this is all about inner non-cellular basement membrane. So we already discussed about all the layers of the insect body wall, that is cuticle, epidermis and basement membrane. Next, what do you mean by endoskeleton? So inward growth of the cuticle. Somewhere the cuticle will grow inward. So the inward growth of the cuticle that provides space for muscle attachment is the endoskeleton. So here this inward invagination is there now that provide space for the muscle attachment. That is the endoskeleton. Endoskeleton, it is of two types. That is apodyme and apophysis. When the hollow invagination is there, then we call such invagination as the apodyme. When a solid invagination is there, here space is less, then we call it as apophysis. Hollow invagination of the body wall is the apodyme, and solid invagination of the body wall is the apophysis. So this is endoskeleton. This is the invagination of exoskeleton, that is body wall. Last slide, that is functions of insect body wall. So it is act as a external armor, mean protective outer shield, just like a armor worn by the soldier. Soldier are wearing that bulletproof jacket, like that structure, body wall. So support and strengthen external organs such as jaws and ovipositors are strengthened by body wall itself. Then after it protects the insect from physical damage, harmful chemicals, parasites, predators and pathogens entry inside the insect body. It also internally protects the vital systems such as foregut, hindgut, trachea. Various systems are then there in our body and insect body also. That systems are protected by this body wall only. It provides space for muscle attachment, maintain body safe, prevent water loss, and help sensory structure to detect environmental cues. Hairs are there now that detect environmental sensation, and it gives cuticular pigmentation and provide body coloration. So this is the role of insect body wall. Good morning, all of you. In this session, we are discussing molting or abdices in insects. See here, this insect is shedding its old cuticle, and this is the remains of the old cuticles. So molting, it is also known as abdices. The periodical process of shedding the old cuticle accompanied by the formation of the new cuticle by the insect is known as molting. This molting process enables the insect to grow. Why? Because the rigid exoskeleton do not expand or allow the insect to grow, and the cuticular remains which are discarded during the process of molting is known as exuvia. See here, egg hatching or eclosion gives first instar larvae. First instar undergoes first molt and gives second instar. Second instar undergoes second molt and gives third instar. Third instar undergoes third molt and gives fourth instar. Fourth instar undergoes fourth molt and gives fifth instar. See here also, after egg hatching, first instar will be formed. First instar undergoes first time molting and gives second instar. Second instar undergoes second time molting and gives third instar. Third instar undergoes third time molding and gives fourth instar. Therefore, the time period between any two molting or the time interval between any two molting is known as stadium, and the form assumed by the insect in any stadium is known as instar. So this is the first instar, second instar, third instar, like that. Frequency of molting. Molting occurs multiple times during the immature stage of the insect, that is in larval and nymphal stage, before they reach the adult stage. The adult stage do not molt further. So molting is there only in immature stage, that is in larvae and nymphal stages. Hormonal control of molting. Molting in insect is regulated by hormones which are secreted from various endocrine glands in insect. See here stepwise control process. Neurosecretory cells. These neurosecretory cells are there in brain. These neurosecretory cells secrete brain hormone, that is PHTH hormone, prothoracicotropic hormone. This prothoracicotropic hormone activates prothoracic gland. Upon activation, this prothoracic gland secrete or release molting hormone. This molting hormone stimulates molting gland which is present in the epidermal layer of the insect body wall. Molting gland secretes molting gel. Later activation of the molting gel will occur. That is, molting gel is activated into the molting fluid with active chitinase and proteases. See here function of molting fluid. This molting fluid digests the old endocuticle, allowing the insect to shed the old cuticle and expand the new one. So steps involved in molting in insects, that is behavioral changes. The larvae which are undergoing molting, they stop feeding, become inactive and shows reduced movement, increased grooming, that is frequently cleaning their body parts, search for a safe and secluded undisturbed spot. This behavior help the insect prepare for vulnerable transition during molting. Epidermal changes. Epidermal cell activity increases, that is, cell size, protein content, and enzyme level in cell increases. Mitotic division occurs, increasing cellular tension. This leads to the loosening of the old cuticle and formation of the new cuticle will occur. Apolysis. Separation of the old cuticle from the epidermis is known as apolysis. It occurs due to increased tension. The cuticle lifts away from the underlying epidermal layer in case of apolysis. Formation of the subcuticular space. A space forms between old cuticle and the epidermis is known as subcuticular space where further molting activity will occur. Secretion of the molting gel. The molting gland of the epidermal layer secrete molting gel into the subcuticular space. Initially, it contains inactive enzymes, that is chitinase and protease. Formation of new epicuticle. A cuticulin layer, that is precursor of epicuticle, is secreted over the epidermis, and this cuticulin protects the underlying cells and a new cuticle from enzymatic digestion. Formation of the procuticle. Procuticle is formed. See here, procuticle formation. That is the orange colored line. Procuticle form below the epicuticle. It consisting of chitin and protein and will later differentiated into exocuticle and endocuticle. This procuticle is later differentiated into exo and endocuticle. Exocuticle is the outer hardened layer and endocuticle is the inner flexible layer. Activation of the molting gel into the molting fluid. Molting gel is activated into the molting fluid where chitinase and protease enzyme become functional. This functional enzyme digests the old endocuticle, but they do not digest exocuticle because which is sclerotized and hard. Up to 90% of the digested materials are recycled to build the new cuticle. Old exocuticle is remain intact and eventually discarded as exuvia and remaining molting fluid is reabsorbed. This is the molting fluid, it is reabsorbed by the cuticle and epidermal layer, new cuticle and epidermal layers. Formation of the wax layer. Pore canals secrete the wax thread that form a protective wax layer. So this is the wax layer. This layer reduces water loss, provide protection against environmental stresses. Next, formation of the cement layer. Dermal glands secrete the cement layer, also known as tectocuticle. This cement layer serves as the outermost sealing layer of the cuticle. Molting process. It involves two steps, that is rupturing of the old cuticle and shedding of the old cuticle. In rupture of old cuticle, insect increase body size. Insect increase body size, insect increase body pressure by intaking air or water and enhancing blood flow. This pressure rupture the old cuticle along with the predetermined line of weakness it called as axial line. It is there in the head region. Setting up the old cuticle, peristaltic movement and the lubrication action of molting fluid help in complete removal of this old cuticle. Along with the outer body covering, the insect also discard lining of the foregut, midgut, foregut, hindgut, and trachea. Post-molt, development of the new cuticular layer. So after molting, exocuticle is developed from the outer region of the procuticle and endocuticle is developed from the inner region of the procuticle and exocuticle undergoes sclerotization via phenolic substances becoming rigid and protective. Endocuticle remains soft and flexible, allowing the body movement in insects. Thank you.